Today I'm going to talk about single-cell application of a single-cell RNA-seq for computational biology. So one aspect of computational biology is looking at disease expression data in disease tissue. Well, it seems failed to respond. The PDF. You might have the error. Starting recording here. Yeah. Okay. And I think it's the RNA-seq community right now have three opportunities. So from blood cells to single cells to single-cell RNA-seq. Probably some of you doing immunology still wonder, is it reasonable? Should we just move on single cell, or should we still just fine-tune our RNA-seq algorithm to look at the bulk sample sequencing? And so I try to provide several rationale. And so we think currently, most of the knowledge of the disease expression profile are based on bulk samples, microarray-based, RNA-seq based. So we sample a tissue, which is a mixed samples. So we make assumption that we look in these average values, and we profile average value. We hope they are really meaningful, and these average values provide us the insight of the key pathogenic pathway and disease mechanism, and we can make a drug out. And the immunology field in the past 20 years, we have to look at these B-cell, T-cell population. In recent 10 years, we start to appreciate they are small fraction of the cells, they are rare population, but they play a key role in immunity. For example, dendritic cells, Treg cells, these are rare population. It is almost impossible to detect their signal in the bulk sample sequence. Then we now have the technology allows us to do the single cell sequencing, and this really can disclose a full picture about molecular events, cell types, and cell states, that we can understand the disease mechanism. For example, what we show here is a schematic map of healthy tissue versus disease tissue. So any sample from the biopsy is a mixed cell type. It could have stroma cell, could have a lymphocyte population, could have a fibroblast. And in the past, we are not able to really deconvolute. We developed a lot of bioinformatics algorithms trying to deconvolute. We probably can deconvolute it a certain way that we know there's a T-cell infiltration, there's a macrophage infiltration, but really the picture is always unclear. And the single-cell sequencing allows us to really compare population versus population between disease and the health tissue. And also we can identify the novel disease pathogenic population. Sometimes the T-cell will be influenced by inflammatory cytokine, and this cytokine will definitely change the feature of the T-cell, and this T-cell become pathogenic. So we want to block these pathways to relieve the disease. And what single-cell RNA-seq can do for drug discovery? In discovery research, so we can ID the cell type. So you have disease tissue from mice, animal model, or from human samples. You want to understand what's going on. You want to compare if your in vivo model we capture the clinical samples. So at least we first of all want to know what type of cell are they. And the second is we will know hidden populations that previously we are not realized, we are not appreciating. Discovering new biomarkers. So it's the first time allow us to really, if we really deep sequencing the samples, truly are able to utilize the clinical features in our gene signature to make the connection. So we can, first time we can identify really, for example, in lupus, we can identify the gene sets really correlate the flare, we can correlate the disease activity scores, which is extremely difficult. So also we can uncover the regulators. So the idea about the single cell is really we look at the gene sets which co-vary together, and they have some biologically meaningful connection between the gene and the genes. So if you really can correlate inflammatory cytokine with some regulator, transcription factor, some other regulatory factors, you really will find a new biology pathway. And we can resolve into temporal pathway. Probably you can find the pathway you previously truly appreciate. So here I just show an example of published by 10 times Genomics. And of course, I put it here just to show you single cell is greater than bulk. Definitely, if you contain more information, you can identify more pathways in single cell RNA-seq data. And here is the one published data, and it's talk about if we just correlate inflammatory cytokine 17, and they really can find some key regulators. Previously, we don't know. And these regulators probably serve a new pathway. We can become druggable. So these pathways, the author proposed it could be druggable. So sometimes there's a connection between metabolism versus inflammation. So we also want to draw a new connection. We want to connect the dots, and some key regulators which we will make a new edge between two genes, which I think is extremely useful tools. So previously I showed several examples about the drug discovery. So how about people will ask the question about how about in late stage biomarker research or biomarker discovery research or even clinical trial? I mean, should we go to single cell RNA-seq or not? So I think this is probably one pretty famous application is talking about gene signature exists in particular population. And this is showing a CD8 T cell, and there's a gene signature which can predict the flare. And of the autoimmune disease such as AAV or such as lupus. So this is the microarray data and using purified population, which in reality, I mean, you have to think about feasibility, how easy you get this sample from the patient, and how easy you. And I think it's extremely difficult. So you collect a large sample, it's a huge effort. And today, if you're using single cell, and we probably can skip the flow sorting. So 10 times genome, and showing perfect example using large amounts of the human PDMC, and they can sequence them, and they can actually not missing major population. So what I want to talk here is single cell RNA-seq really can help to identify, discovery, or monitor cell-specific gene signature. If the signature serves as a biomarker, we can really put it into the clinical research. And then I also will propose that if you do clinical research, if sometimes fact sorting isn't feasible, it's hard to do, and maybe we'll think using single cell RNA-seq to digital sorting the cells. So this is our motivation that why we want to work with Omicsoft together, and I think that John mentioned previously. Omicsoft will launch their new product, single cell RNA-seq or single cell length, which focus on the single cell biology data. And so we basically were lucky to have a preview of data. We use parts of the data and to understand the technology. And so in December, I think the data will be launched. It includes 35 targets and 13 key samples involved. So they provide us a lot of metadata, a lot of QC data, and sometimes I'm a data dump person. I still like to have all the data in our studio, our session, and play with it. And in this land, currently we have 50% of the human data sets and the 50% of coming from mice. And this currently includes the technology, including C1, fact sorting, some lab manually pick it. So I think it is the early stage of single-cell RNA-seq. The whole field has still compare different methodology. And I think the different methodology all depend on particular experiment and the phase of the methodology. And so we choose some immunology relevant data, and we look at I think there's several key features provided by Omicsoft lens. Some QC data they did for us. So one of the key 17 data sets they have, I need to look at alignment rate. I think it's quite important. So we consider 70% of alignment is reasonable in currently. We also look at types of transcripts, because one challenge of the single-cell RNA-seq is that it's all the population we need to ID them. So we need to know what they are. You know, it's just like each cell has their own driver license or social security number. We just want to know who they are, and using our informatics method. But sometimes there's a contamination in the population. So I heard about someone talk about mitochondrial burst in their tissue. In the samples, certain cells express a lot of mitochondrial RNA. And when we discuss with John, so we think it is probably the cell is not meaningful. It's artificial. And also sometimes we really want to compare, spiking and versus RNA. If you a lot of spiking, but then real RNA-seq, so probably you want to remove this sample. And also we look at a sequence depth. At least, I'm not trying to set a threshold, but just if you only have thousands of leads, probably it doesn't make sense to go further. And gene coverage, so that's Omicsoft has made the definition of a coverage means the minimal number of the genes are expressed in particular cells. So which means how many genes can you recover compared to the whole genome. And also we think it's important in the near future, I encourage everyone just try to at least put the RTC spiking your samples, run even in single cell RNA-seq experiment. So there's several features we believe it's depend on us who use the land data, because sometimes it's hard for Omicsoft or data curator to really identify the issue of the data. And I will show example later on. And for example, minimal percentage of the cell express particular gene. If you're interested in one gene, but you say 10% of your sample expressed it, how would you decide? Is this meaningful? Is this artificial? So if you have a mixed PBNC, probably 1% of cell is extremely important, because they could be plasmacytoid cells play extremely role in immunity. But if you have a pure T cell population, if 1%, maybe it's an artificial one. And also, I'm extremely careful about contamination and double edge. So the author who loads their data in public space already carefully curate data sets, because they really don't want to put all the data, share everyone, then you found that that's a lot of problems. So it's kind of shameful for them. So they oftentimes they clean the data. But sometimes they overlook the problem too. So we need to be really, really careful about that. And so I just walked you through several QC and Omicsoft provider and how we use it. So as I mentioned, so there's a mapping rate. and we just plot and so we're using T17 and mice data and really plot the mapping rate. And we see there's a one, so x-axis represents the mapping rate, and the y-axis represents how many cells. So majority of cell has at least 60%, 70% of mapping rate in this data set. Also, we look at the gene coverage. So in this graphic, x-axis represents the coverage and the y is the sequence. So which means if you look at it here, so there could be 150 samples or 150 cells. We can find 10% of their genes has FPK value greater than 10. And we also look at the type of the transcripts. I think it is extremely important because this is helping to find the outlier data. And so we look at mitochondrial no burst and the no microRNA and no link RNA. And we have a small population of immunoglobulin genes, which is strange in T cell. And I explain why. So basically, there's a lot of features you can play with it, and we basically we put the insertion size and correlate with the mapping rate. Apparently, there's something showing up, and then you will see high insertion size and correlated with a good mapping rate. And then we really go back to data. We found out they merge different batches. So a single cell RNA-seq batch effect is a major issue. So I think that then it's great. We know the QC and we kind of these reasonable data sets. We want to remove some low quality and data. We put them together. This is the common way we do analysis. We look at the most available genes from these samples. So here's the plot for average expression versus dispersion. So you can find the available gene, and the PCA plot is another very powerful method, and many people use it. In this setting, we surprised at the beginning. It surprisingly behaves really well. How come in T cell T17 I have distinct population working so well? Oh wow, we found it. We hit 70 cells. Then we further go detail. Go to really look at all these gene string profile. We found there's a contamination from B cell and M cell. So then we really remove the cells, and further we continue to do the PCA and continues. So I think that's extremely important. You should really be careful about what the cell you are doing analysis, because if you rely on all the mapping pipeline, you rely on some PCA analysis, the first principle component, you probably get the wrong answer. And we went back to the paper. I think that also overlooked it. So there's some table, their computation table was totally wrong because they saw when type of T17 they should CD14, CD19, it totally isn't the marker of the T cells. So then we find very interesting. Then we further after we make sure we really deal with T17 cells, and we deal with T17 cell from the central nervous system and T17 just from lymph node. Then we really compare these two types of cells to see what's the difference. We also utilize experiment derived gene sets on some gene signature database. So we know certain genes, they are really connected to the acknowledgement pathway, which means with certain cytokine, inflammatory cytokine cause a series of a gene upregulation. And this gene upregulation, we believe is a set of genes is related to the pathology. So we find it interesting, these pathogenic genes, which due to the inflammation, pretty much expressed greater than 50% of the Th17 cell from the central nervous system. But really not from the lymph node. So apparently the Th17 cell in different tissue are different. And then we try to so this plot is we just mimic the flow cytometry data. Basically, so if you look at a particular one gene, you can plot this fact flag plot to really show in in certain genes in central nervous system. Highly enriched, but not in the no, so we do experiment for you generate a hypothesis to say certain pathway may link to disease. So then you do in vitro experiment, then you identify many genes that relate to this pathway. So then we have internal annotation, these genes relate to certain pathways. Of course, if you knew it, or if certain pathways are already in GeneSense, or GSEA analysis database, there's a multiple way. So, let me summarize what I'm trying to say. Okay, so I think for single-cell RNA-seq, we really, I think it's a very exciting, very powerful technique, and QC data from the Omicsoft land, single-cell land, is very, provide us lots of information, and we can really diagnose. Without this data, I couldn't quickly, so when I read a paper, I couldn't really quickly understand what they did, what the carry-outs out there, what we can use. And also, be really, really careful to remove not only low-quality data, also contamination. It's really not sure, each cell's ID, really make sure if that's really the cell you think it is. And transcriptome profile of the single-cell RNA-seq is powerful. So especially we focus on to analyze the gene reflecting pathological pathways. And so I want to hear really my final message to support Omicsoft to further develop the single cell. So then incorporate the new feature like cell cycle stage assignment, because there's some T cell population. So you activate T cell, and the T cell probably not going synchronized, there's unsynchronized the cell, and you want to you want to really dissect them out. This is impossible to do in the bottom RNA-seq. And normalization method, how we call the differentially expressed genes, and how to automatically identify cell population, and how to combine the power of the ERCC with your samples. And I think at this early time, exciting time for single cell RNA-seq. There's a lot of things we can learn using the published data. We know the advantage and disadvantage of the technology. And finally, I would say, so I think this effort, we closely work with the John and Gary to the Omicsoft. And so I also want to thank, so we in BI and computational biology, closely work with immunology. Oftentimes, with coming back for a discussion how we utilize the data with therapeutic area scientists. So I also want to thank Kai. We always explore different R package, and you always really support for this talk. And I also propose that we should have some consortium effort included in RNA-seq. So in a session of the single cell RNA-seq, to see if it's all the industry's interest, should do that. I think I'll give you some questions about. Yes, I think it's a particular case. When you talk about cell cycle, you talk about T cell and activated T cell, proliferated T cells. And currently, there's only probably one data set out there. I think I think it's nice because we know it's true because there's a UK enzyme. So my experience, UK enzyme, particular cell cycle, using prior knowledge to really define. So the value is it gets you. I think it's a more statistical analysis, depending on what you get from it. I think it's useful, but without it, I mean, you still get lots of meaningful results because just, for example, if you correlate the particular gene expression with cell cycles, you can remove lots of genes, and the remaining genes could be interesting. Sometimes it's just a confounding factor. Rather than you really want to understand, because the T cell, you say, are you really interested in cell cycle, particular cell cycle correlate the genes? I do not know. So it's just about how you use this data. So I think that the good thing is, if you design the cell cycle to the cell, and you also correlate certain genes with the cell cycle, it depends on how you use it. I think I answer your question. But it's only with data I'm very careful about to use all. I mean, if you work on cancer, it's great for the T cell. I know it depends on your experiment. Your experiment is really has a lot of cell cycles and things like that. You probably will say this is a component I want to take a look at it, but usually it is you say I want to remove this confounding effect. Then using statistical analysis to utilize and to see the picture that I haven't seen. So it's translates TH2 data in the end, which showing certain transcription factor to drive the TH2 polarization to TH1 or probably TH22 or something like that. But it all depends on what. So we have to. So in single cell, we're still deconvoluting information. Doesn't mean we can truly know every gene what they really do. But it's much better than previously, just like almost similar as why you want to do WGCNA analysis. Why you have to spend so much time collect hundreds of samples in the bulk. So I think it depends on purpose. Yeah, well, if you look at it, you can 10 times genome. I mean, the company, I just showed a picture that they can decouple TH cells and using large sample size, right? So in some data, you still need to focus on depending on how deep you want to go. So if you really care, so I show example TH17. So of course, if you're doing PBNC, of course, it's be very, very difficult to deconvolute to see all the information about small fraction of cells still. So you need to really estimate, let's say, it doesn't mean you do all PBNC, solve the other question. So if you really focus on particular subsets, you still want to partially enriched. Partially, let's say I want to remove this confounding cell type neutrophils, I don't want it, then the remaining cell may be capable. So if you want a T cell, it's better you can just try. You can try the preliminary experiment doing the T cell to see if you can really find what you're looking for. It's really you really want to estimate if it's doable, then you apply it.
So I don't want to cut off more questions, but I'll make sure for the rest of the good chance to talk. No, it's okay. So if we end up having more time, if we may, if there's additional questions.